Hi dear students, let's take next section in the general physiology. We are discussing now the concepts and principles in the general physiology, and we will mainly focus on the ICF and ECF. We discussed in the previous section the body fluid compartments. So I'm going to include a few more points related to the body fluids, ICF and ECF. And then we will discuss the homeostatic mechanisms. What all questions have been asked in the past, recent past related to this? For example, a question like which of the following is the most efficient homeostatic regulatory mechanism in the body? And the options given are baroreceptor mechanism in blood pressure regulation, temperature regulation by hypothalamus, pH regulation in the body, and kidney body fluids mechanism in the regulation of blood pressure. So most efficient mechanism, of course, is this in this case kidney body fluids mechanism. Why? We will see that in the discussion. Apart from that, the other questions, they can ask you to calculate the gain, which is the indicator of efficiency. We will see how we calculate the gain. Then the other question that was asked recently was which of the following is the positive feed forward mechanism? Let me drop the word positive here. Let me not complicate the things at the start. The question was which of the following is the feed forward regulatory mechanism, and the options given were baroreflex mechanism in blood pressure regulation, then oxytocin and its action in parturition, then cephalic phase of the gastric acid secretion, and osmoregulation by hypothalamus. So very often in the first year people read positive feedback and negative feedback, and they confuse positive feedback with feed forward. The question was specifically on feed forward, and the answer was cephalic phase of the gastric acid secretion. Why? We are all going to discuss all of this in this chapter. So let's begin with the discussion on the body fluids. We have done the body fluid compartments in the previous section, measuring the volumes of those fluids. Now I need to add some conceptual fuel to the previous discussion. So let's see what we are going to discuss here. Let's say this is a cell and the intracellular fluid inside the cells, and the cells are surrounded by the ECF. ECF has a high concentration of sodium, 141 milliequivalents per liter. So if you were to be asked the major cation in the ECF it's the sodium. Sodium concentration in the ICF, intracellular fluid, is low, and it's quite understandable. You know why? Because sodium is osmotically active. It pulls water towards it by osmosis, and therefore if sodium remains high inside the cells, then it will pull water into the cells. All the cells will swell and burst, and therefore we will see later that we have specific mechanisms to extrude this sodium out of the cell. Okay, but remember, for now this is the sodium. Coming to the potassium. Potassium is in a high concentration inside the cell, 145 milliequivalents per liter, and it's low on the outside. Potassium is always pushed into the cells. So remember, major cation inside the cells, major cation in the ICF is potassium. Okay, so major cation in the ECF, sodium; major cation in the ICF, potassium. So far as the anion is concerned, the ECF is rich or high concentration of chloride. Chloride is in high concentration in the ECF, whereas ICF has the anions in the form of the intracellular proteins, and that is what we are going to discuss now. But this is our starting point. Okay, now based on this, I need to tell you three important points. 3 points. Okay, first point, and let me tell you this in the form of an MCQ. There was an MCQ in one exam in the INI. The question was, which of the following body fluids, some body fluids have been mentioned, which of them is actually an ECF, but it more resembles like ICF. It's actually a part of extracellular fluid, but it more resembles the intracellular fluid. And the options given were synovial fluid, intraocular fluid, CSF, and endolymph. You're taking the guess on that? The guess? Endolymph. Yes, that's my first point. Just a small conceptual point is that about the endolymph. Look, we have mentioned that potassium is low in concentration in the ECF. There is only one exception, and that's endolymph. You know, in our labyrinth in the inner ear, so one part of it is vestibular apparatus, the other one is the cochlea. The receptor cells are the hair cells. And those hair cells are surrounded by a fluid called as endolymph. So in short, endolymph is a fluid inside our semicircular canals and labyrinthine organ or vestibular apparatus. This fluid is having a high concentration of potassium. See, this is an extracellular fluid, okay? And the point that I'm trying to make is that this fluid has the ECF composition because it is an ECF, it is a part of extracellular fluid, and over and above that, it has excess potassium ions. Okay, so please remember this for now. In this sense, endolymph, although it is ECF, but it resembles the extracellular fluid in the sense that it has a high concentration of potassium. Okay, since it is an exception, I thought I'll just mention it at this particular point, and since already MCQs have been asked on this. Okay, so that was the first point. Second point which I want to highlight conceptually is related to the equilibrium, commonly called as Donan equilibrium or Gibbs-Donan relationship, Gibbs-Donan equilibrium. Look, and again it's a concept that you need to understand clearly. There are diffusible cations and anions in both these compartments, the ICF and ECF. Both these fluids, both these compartments, potassium, calcium, chloride, magnesium, lots of diffusible cations and anions are present in both the fluids. Okay, but in addition to these, there are impermeant or non-diffusible anions in one of the two compartments, and that compartment is intracellular fluid. The concept is this: there are lots of diffusible cations and anions in both the fluids. Okay, so as I mentioned, sodium, potassium, calcium, chloride, bicarbonate, etc. And by diffusible, we mean that they can diffuse through the membrane. In addition to these, there are non-diffusible anions in one of the two compartments. Which is that compartment? The ICF. So let me first say what those anions are. There are intracellular proteins. Okay, proteins are anions, you know it already. And phosphate. You know, inside the cells there are high energy phosphate. There is high energy phosphate. So phosphate and proteins, these are the anions which are trapped inside the cell, and they can't diffuse out, they can't leak out of the cell. So we have anions inside the cell which can't diffuse through the membrane. Which means what? Which means there are excess anions inside the cells. there are excess anions in the ICF. That is our starting point. Look, you must understand clearly what I am trying to establish. There is law of electroneutrality, okay, the law of equilibrium or the law of electroneutrality. So the number of cations and anions should be equal on either side of the membrane or in the neighboring compartments. There should be a certain equilibrium. Now what is happening is, to achieve that law of electron neutrality, or to achieve that equilibrium, to maintain that electroneutrality, the diffusible cations and anions are moving through the membrane, diffusing through the membrane to maintain that electroneutrality, so that the number of cations and anions is equal on either side of the membrane or in both compartments. Now the point is, these anions which I mentioned just now, they cannot diffuse through the membrane. They cannot take part in this process of achieving the equilibrium. And therefore what happens is, now this point is important. Conceptually, you should make a note of this, that our starting point is that there are excess anions in one compartment. Excess anions inside the cells. Okay, you will have to remember this forever now as your concept, that there are excess anions inside the cells, and all the cells without any exception. Now because this is our starting point, and as I mentioned, there is a law of electroneutrality, and here I say proteins and phosphate inside the cell. So excess anions is our starting point because they cannot diffuse through the membrane. So what is happening in every cell and continuously is, cations are being pulled into the cell towards these anions because there are excess anions. So electroneutrality should be maintained. So equal number of cations also should be there. They should come in. And therefore, what I want you to understand is, cations are pulled in to the cell. Which is that cation that is being pulled in? Sodium. Naturally, because sodium is excess in the ECF, and therefore, please remember that sodium is the cation which is constantly leaking into all the cells of the body. And continuously, this is happening continuously. Why is sodium continuously diffusing into the cells, all cells of the body? To attempt to reach the equilibrium. It is attempting to reach that electroneutrality. Excess anions are there, and therefore sodium is also coming in from outside. It is making an attempt to reach that electroneutrality. Fair enough. At this point, you can remember this much. Sodium does not stay. It comes in, but it does not stay. Okay, it's only attempting to stay. It's not going to stay inside the cells. What happens to it? We will see that later. Already we have said that sodium has to remain low intracellularly. Okay, so this is going to be thrown out. Please keep that in mind. However, conceptually speaking, I need you to take this point as a second point. Excess anions inside the cells. And therefore, the cation, that is sodium, is constantly being pulled into the cells. The third conceptual point that I wish to mention here is this, about the concentration gradients and electrical gradients. Concentration gradients and electrical gradients. Another important concept which you will need, particularly for the excitable cells in the body, the muscle, cardiac muscle, smooth muscle, skeletal muscle, nerves. You will need these concepts. So focus on the third concept. There is diffusion of ions. For instance, I mentioned sodium. Sodium, its concentration is 141 milliequivalents per liter on the outside and 14 milliequivalents per liter on the inside. So in the case of sodium, its concentration gradient is pulling the sodium into the cell. It's causing the diffusion of sodium into the cell. That's by concentration gradient. Yeah, you know, diffusion occurs from high to low concentration, and so sodium's concentration gradient will pull the sodium into the cell. That's one point for sodium. But what about the electrical gradient? Remember, we saw it just some time back that there are excess anions inside the cells, okay, and that means relatively excess cations on the outside. We are talking in the relative terms here, relative to each other, okay. So when I say excess anions, it's in relation to the ECF, right? So you see, there are excess cations on the outside, and I mean high concentration of cations on the outside, and relatively low cations and high anions, high concentration of anions inside the cell. So this creates an electrical gradient. Look, we are talking in terms of cations and anions, means the charges, electrical charges, and this creates an electrical gradient. So this electrical gradient with excess cations on the outside of cell and low cations and high anions inside the cell causes any cation like a sodium to move in from outside to inside, okay. So this time I will use the expression sodium will diffuse from high number of positive charges to low number of positive charges, okay. Diffusion occurs due to concentration gradient, that's this 141:14. Diffusion also occurs because of the electrical gradient, that is from high number of positive charges to low number of positive charges a cation will diffuse, and that's called as diffusion by electrical gradient. So a cation like sodium will diffuse from outside to inside by electrical gradient, I beg your pardon, by electrical gradient, which means what? Which means sodium will always diffuse in to the cell. Sodium will always, whenever we say diffusion of sodium, it has to occur from outside to inside, because both the forces, the concentration gradient and the electrical gradient, will pull the sodium from outside to inside. But my point is, I stretch it a little further, and my point is, when I say concentration gradient, I mean the concentrations of that particular ion. Sodium 141 milliequivalents on the outside and 14 on the inside. So concentration gradient means concentrations of that particular ion. And when I say electrical gradient is created by all the cations and anions in those two compartments, okay. Electrical gradient, when I say excess cations on the outside and low cations on the inside and relatively high anions on the inside, it is created by all the cations and anions in those fluids. So when I say excess cations are there in the ECF, it's I'm talking about all the cations put together in the ECF, yeah, sodium, potassium, calcium, magnesium. So all cations put together in the ECF and all cations put together in the ICF, if you compare, there will be more cations on the outside. So remember, I repeat once again, I know it's going to be repetitive, but this is very important, lots of concepts based on this. So remember, when I say concentration gradient, I mean concentrations of that particular ion. When I say electrical gradient is created by all the cations and anions in those fluids, right? But it's not yet over, okay? So sodium will always diffuse in. Now coming to the potassium. This will be interesting. What is the concentration gradient for potassium? Concentration gradient for potassium, potassium is high on the inside, 145 milliequivalents per liter, and low on the outside. In the ECF it's low, so potassium. So by concentration gradient, potassium will diffuse outward from inside to outside, okay? But then what about the electrical gradient? Think about it. Electrical gradient. Ooh, electrical gradient will pull the potassium from outside to inside. Remember, I just now told you about the sodium, that there are high number of cations on the outside and relatively low number of cations on the inside, and excess anions in the ICF. So that means, and I mentioned it, that any cation will always diffuse from outside to inside by virtue of electrical gradient, the force created by the electrical gradient. So potassium is a cation, and therefore potassium will have another force for diffusion, and that is from outside to inside by electrical gradient. Remember, when I said sodium diffusion will always be from outside to inside. When I said potassium can diffuse in or out. It will diffuse out by concentration gradient, and it can also diffuse inward from outside to inside by electrical gradient. So in summary, I want to just re-emphasize 2 points again. One is, this is just a repetition again, but let's just keep this consolidated. First point, when you think of concentration gradient, you are talking about the concentrations of that individual ion. And when you say electrical gradient, you are talking about the electrical charges created by all the cations and anions in those particular fluids, okay? So when I say excess cations, I am including sodium, potassium, calcium, magnesium, all of them put together, okay? That was one point. And the second point, which I had to highlight at the end of this section, is that sodium will always diffuse in from outside to inside. Well, and I also mentioned to you that sodium diffuses in all the time because there are excess anions, yeah? So sodium will always diffuse in because both the forces, concentration gradient of its own and the electrical gradient for the cell, both of them are pulling the cation in. In case of potassium, it will move out by concentration gradient, but it can move in by electrical gradient. So therefore, I am ending this particular section by saying that what is the concept of equilibrium? What's the concept of equilibrium? Okay, now consider this: potassium moving from inside to outside by concentration gradient, and that will have certain magnitude, and potassium going from outside to inside by electrical gradient, that will have certain magnitude, that will have certain flow rate. These two forces, they are opposite to each other. When they are equal, remember, when these two forces are equal and opposite already they are, but when they become equal, the potassium will reach equilibrium, okay? So please remember the concept of equilibrium. We are coming to the end of this section. Many students, they tend to think from first year equilibrium, they just think like it's equal, means something 145 was there on the inside and 4 on the outside, so some 75 have traveled from inside to outside, so 75 on both sides, and that's equilibrium. That is a very wrong concept that many students have from first year. That's not equilibrium, that's equal. I'm talking about equilibrium. When will an ion reach equilibrium? When the two forces which are acting on it, they are equal and opposite forces balanced out, which means what? Which means there is no net movement of that ion. Net movement is not occurring. Whatever amount is going out, equal amount is coming back in. So two forces have balanced out each other. No net movement of the ion. That is the concept of equilibrium. I tell you, equilibrium is a hypothetical concept, means it's a theoretical concept, it's a hypothetical concept. It does not reach, ions do not reach equilibrium because they are constantly moving in, moving out by certain forces. So I need you to understand even this part, that equilibrium is a hypothetical or theoretical concept. What will happen when the two forces become equal and opposite? That's a rarity. It generally does not happen that way because lots of other forces are happening in those fluids. Okay, let me also add a comparison of the two terms. The two terms which are commonly used are equilibrium and steady state conditions. You will come across these terms often in physiology. Let's take the example so that you will understand properly. When you say equilibrium, the example is, we can take the example of potassium. Potassium concentration gradient is driving it from inside to outside, and electrical gradient is driving the potassium from outside to inside. Yeah. So concentration gradient, electrical gradient, when these two forces balance out each other, there is no net movement of potassium, and potassium has reached equilibrium. So that's the example. A steady state condition, there are lots of examples. For example, when blood glucose is maintained at 100 milligram percent, it's held constant at 100 milligram percent, we say this is a steady state condition. Or when blood pressure is maintained at let's say 120 millimeters of mercury, it's a steady state condition. So steady state condition is a reality. Okay, it does happen in the body. Equilibrium is a relatively hypothetical condition or theoretical condition. If that happens, these are the words for equilibrium. Okay, let's compare the two. Equilibrium means it happens between the two neighboring compartments. When you say there is an equilibrium, you are essentially talking about two neighboring compartments. Equilibrium happens between the two neighboring compartments. But if I say steady state, not necessarily there will be two neighboring compartments. It may be a single compartment, or it may be several adjacent compartments. So it may be single or multiple compartments we may be talking about in case of steady state. Equilibrium also involves two equal but opposite forces which are balanced out. When you say equilibrium, two equal but opposite forces have balanced out. In the case of steady state, not necessarily that's the case. Look, for example, to maintain the blood glucose at 100 milligram percent, there are several forces and they are not necessarily equal and opposite. Okay. Third and the most critical point, this will differentiate between the two. Equilibrium, attaining and maintaining the equilibrium does not need ATP consumption. Yes, we took the example of potassium. It does not require ATP consumption because it's totally based on the diffusion. diffusion by concentration gradient and electrical gradient. Diffusion does not consume any ATPs. So no ATP is required to attain or maintain equilibrium. Whereas to attain and maintain the steady state, ATP breakdown will be necessary. ATP consumption will be necessary. For example, blood glucose. Insulin is one factor, and just for the sake of release of insulin also ATPs are consumed. So to attain the steady state, ATPs are consumed. And the one more point is equilibrium is relatively short-lived condition, whereas steady state condition is a relatively long-term condition. Look, I mentioned to you already that the equilibrium is almost a hypothetical condition. What do I mean? I mean that imagine at some point potassium has reached equilibrium, means both forces are equal and there is no net movement of potassium. But then it will be very short-lived because there will be some other forces acting, like cations or anions changing their concentrations, and therefore electrical gradient will get altered, and therefore those two forces will not remain equal. So equilibrium will be disturbed. It's sometimes called as disequilibrium. However, but it's a short-lived condition. Even if it was achieved, it will be a short-lived condition. Steady state is relatively long-term. It's maintained like that for quite some duration. So that's a comparison between two very commonly used terminologies about the concepts in physiology. Let's see first two definitions. One is myelointerior, the internal environment of the body. I am making a simplistic pronunciation of this word, myelointerior. It's written like this: internal environment of the body. And for the body, the myelointerior is nothing but ECF, extracellular fluid, because it's the myelu for the cells. Cells are surrounded by the ECF. Cells are bathed in ECF, and therefore extracellular fluid is the myelu for the cells, and that's the environment, internal environment of the body. A very renowned physiologist by the name Claude Bernard is credited with coining of this term, myelointerior. And the other term is homeostasis, maintaining the constancy or stability in the myelointerior. So that's another term, stability of the myelointerior. That's called as homeostasis. Another very renowned physician by the name Walter F. Cannon is credited with coining this term, homeostasis. So we are going to discuss about the homeostasis and its principles. The point is that myelointerior, the extracellular fluid, has lots of parameters in it. Those parameters are called as controlled variables. What are they? The nutrients, glucose, oxygen levels, carbon dioxide levels, pH, temperature, osmolarity, lots of them. And only when these parameters are held constant at their normal range or within their normal range, only then the cells can function optimally. So maintaining that constancy for all the parameters and thereby maintaining the stability in the myelointerior is defined as the homeostasis. So what are the various mechanisms by which homeostasis is achieved? Let's first see the model on which the homeostatic principles are based. The schematic design, the schematic model for homeostatic mechanisms in the body. First of all, before I start this, let me tell you the ultimate goal for all the body systems. The ultimate goal for them is homeostasis. I mean, for instance, cardiorespiratory system. It regulates what? These two systems, they would regulate oxygen and carbon dioxide levels. Then digestive system. It will maintain the nutrient levels so that there is a steady supply of nutrients to the cells. Kidney, excretory system. It will wash out all the excretory products and thereby they don't get pile up in the ECF. So every system has ultimately the same goal. Even something like musculoskeletal system. What is its contribution in homeostasis? Suppose blood glucose levels drop. What is expected? You will walk to the kitchen and eat something and the blood glucose level will return to normal. So this walking to the kitchen and then taking something to eat, this is achieved by the musculoskeletal system. So, you know, even this system has played its part in the homeostasis. And of course, there are two systems which are the regulatory systems, the nervous system and the endocrine systems. They regulate the homeostatic contributions of all other systems. So, for all these parameters which are regulated by the various systems, there is a schematic model for homeostatic regulatory mechanisms. Let's see what that model is. In the myelu interior, there are controlled variables. Each parameter in the myelu is called as controlled variable. And I mean, pH is a controlled variable, osmolarity another controlled variable, temperature another controlled variable. So these parameters are called as controlled variables. For each of the controlled variable, there is a sensor specifically designed for that control variable. So sensor will sense the value of the control variable constantly, and accordingly it will send the signals to the controller. For each control variable, there will be a control variable which will be monitoring those values. For example, blood pressure is a control variable. Then baroreceptors are the sensors and they will send the signal to the vasomotor center, which is the controller. The controller then will compare this value with its set point. Already there is a set point which is fed to the controller. So this value which is now sent to the controller, it will compare the value with the set point, and accordingly, if required, the controller will take the action via the effector organs. Effector organ or effector organs. So that's the schematic design for all the homeostatic regulatory mechanisms. Having said this, let me now move forward and let's now see that there are basically two types of regulatory mechanisms. One is called as feed forward and the other one is feedback. So these are the basic two types of regulatory mechanisms, and even in the feedback there is negative feedback and positive feedback. Even in feed forward we would have negative feed forward and positive feed forward. So let's try to understand what these mechanisms are. Look, if there is a disturbance, like blood pressure has changed already, then it is sensed by the baroreceptors, signal sent to the controller, and then the controller takes the action. It would be called as a feedback mechanism. It's a feedback which is coming from the myelu, and then the controller is taking the action, and whatever desired action, bringing the control variable back to its normal level. On the other hand, if the controller anticipates that there is going to be a disturbance, and even before the disturbance actually happens, the controller takes the desired action, then it would be called as feed forward. You understand? Feedback is when disturbance occurs and then the controller will take the action. Feed forward means controller anticipates that a certain disturbance is likely to happen, and it takes the action even before that change has actually happened, that disturbance has actually happened. The controller will take the action. That's feed forward. So it's quite obvious to understand that feed forward will be a touch better as compared to feedback. Why? Because it will save time. It will save time. There will be no time lag. In the case of feedback, disturbance will happen, then it will be sent to the controller, then controller takes the action. There will be a time lag, time lapse. In the feed forward, no time lapse. In anticipation of the disturbance, the action has been taken. So that's basically, I'll give you the example. Just upon seeing the food, there is reflex salivation or reflex secretion of gastric juice. Even before the food has gone into the stomach, there is secretion in the gastric juice. This is a classic feed forward regulation, because this is now, it's anticipated that the food is going to go into the stomach, and therefore to prepare the stomach for that, the gastric juice secretion starts, and therefore that's feed forward classically. Okay, let's now try to understand these mechanisms one by one. Let's summarize what I said just now. Feed forward means controller anticipates a change even before that change has occurred. It means the same, anticipating the change, and then takes the desired action. And therefore, what is the benefit of this? There is no time lag, or there is no time lapse. Controller has anticipated the disturbance. Let's see the examples. The example which I mentioned just now, cephalic phase of the gastric acid secretion. And if you are already aware of this, we experience this as well sometimes. Just on seeing the food or thinking about the food or smell of the food, and there is either reflex salivary secretion or reflex gastric acid secretion. So that's called a cephalic phase of the gastric acid secretion. Salivary, which I am mentioning, is a type of conditioned reflex. We will see that in the digestive system. Okay, so that's one. Apart from that, any other example? Yeah, increased respiratory drive. Increased respiratory drive just at the start of exercise. This is feed forward. Look, when you start the exercise, muscular exercise, muscles will need extra oxygen. In anticipation of that, when the exercise begins, just at the start of exercise, the ventilatory drive will increase in anticipation of the fact that muscles now will need extra oxygen and they will generate extra carbon dioxide. So these are the feed forward mechanisms. Now let's come to the classic feedback mechanisms. Feedback homeostatic mechanisms. And as I mentioned, there are two types: negative feedback and positive feedback. First of all, what is a feedback? Change occurs, or let's say disturbance occurs in the milieu, in the control variable. The control variable is changed. In fact, it is fed back to the controller, and then controller takes the action, desired action. So this is called as feedback. And what is the, let's say, disadvantage, or let's say fallout of this? There is a time lag. Means for some amount of time the disturbance will stay. You understand this? Disturbance has happened, then the signaling will occur for the controller, and then the controller will try to rectify the change. So for some time the disturbance will stay. In the feed forward, there was no time lag because controller acted beforehand. What are the two types? Okay. Negative feedback. Negative feedback is the one in which when change occurs, disturbance occurs, controller does the opposite. Controller does the opposite. So controller negates the change. Controller negates the change or cancels out the change, so that the control variable, the parameter, is brought back to its original level, normal level. That's called as negative feedback. I mean, if the blood pressure has increased, the controller will bring it back to normal, or decrease it and bring it back to normal. Or if the blood pressure has decreased, controller will act in the opposite. It will increase the blood pressure and brought back to normal. So that's a negative feedback. Controller does the opposite. And because of this, that the controller does the opposite, the error is minimized. I'll tell you what is error. We are going to talk about it. But for now, just remember, in the case of positive feedback, error will be amplified, magnified in the case of positive feedback. Remember this. And in the negative feedback, error is going to be minimized. So what is this error? Let's try to understand now. What is so-called efficiency of a homeostatic regulatory mechanism? We will discuss about error in the context of efficiency of a negative feedback mechanism or homeostatic negative feedback mechanism. So we can measure the efficiency. How efficient is a negative feedback mechanism to bring the controlled variable back to its normal level? Okay. The efficiency of any feedback mechanism is measured. Look, can there be a measurement of efficiency? Yes, the efficiency can be measured in terms of gain of a negative control mechanism or negative feedback mechanism. Gain of the particular mechanism. This is not being used the way we use it normally in our normal conversation. What's the gain for me? No. This term is generally used in engineering. So gain, first of all, keep this in mind, that gain is the measure of efficiency of a negative feedback mechanism. And gain is equal to correction applied by the negative feedback mechanism upon residual error. Correction applied upon residual error. This will be required for you to solve the MCQs. They may give you the data and ask you to calculate which one is the most efficient system. Let's take an example to understand this. Let's say blood pressure is at its normal value 100 mm of Hg. The mean blood pressure is about 100 mm of Hg. Some disturbance happens and the blood pressure increases to 150 mm of Hg. So because of the disturbance, error has been created. Now this error signal will be fed to the controller and then controller will take the action. This is a negative feedback. So the error will be negated, cancelled out, and the control variable, the value is brought back to its original level. That's a classic negative feedback. But let's say, now this is an example. This is just an example to illustrate the formula. Let's say this particular BP regulating system brought the blood pressure up to 110 mm of Hg. It did not go all the way back to 100, okay? From 150, it came and stayed at 110. So this is the correction that was applied from 150 back to 110. But as you can see, there is some error which is still remaining. Value has not gone exactly back to 100. So this remaining error, or it's called as a residual error. Now you can see and tell me what is the gain in this particular example. Correction applied upon residual error. Can you calculate? Well, it's easy now. Correction applied was of 40 mm of Hg, and the error that was still remaining, the residual error, that is 10 mm of Hg, and therefore the gain in this particular example is plus 4. Now plus or minus do not indicate the magnitude, it just indicates the direction of the final change. So let's further describe this. Higher the gain, more efficient is the system. Obviously, that's quite easy to understand, because higher gain means, it would mean that the correction applied would be large and residual error will be very small, doesn't it? Denominator will be small, numerator will be very high. So that means that the system is very efficient. It is almost completely correcting the disturbance and leaving very small error. So that will be a very efficient system. Yeah, so higher the gain, more efficient will be the system. And therefore, let me tell you some of the gains of the homeostatic feedback mechanisms. It is said that the kidney body fluids mechanism in regulation of blood pressure is said to be the most efficient negative feedback mechanism or homeostatic mechanism, because it is said to have infinite gain. Infinite gain means zero error, it leaves zero error, denominator will be zero. Apart from that, some systems with a higher gain, temperature regulating system, one of the most efficient systems in the body. Temperature regulating system has got a gain of 34, very efficient system, whereas baroreceptor system has got a gain of 2 to 4, and temperature regulating system has got a gain of 34 to 36. So the values given by different authors in different books. Now these are the gains. So temperature regulating system has got a very high gain, and baroreceptor system lower gain, less efficient system. That was the idea of efficient mechanisms in the body and their measure of their efficiencies. Finally, let's also try to understand the other mechanism, positive feedback mechanism. Positive feedback means the controller does the same. Means if a change has happened, controller will act in the same direction. Controller does the same. Remember, in negative feedback, controller does the opposite. Means a change has happened, controller brings it back to its original value. In this, controller will perform the action in the same direction of the change. So what happens is, for example, A leads to B, B leads to again A, and again A leads to B. More and more of the same goes on happening. That's positive feedback. More and more of the same goes on happening. So this is sometimes also called as vicious cycle. Because yes, vicious, while, because if more and more of the same goes on happening, the control variable will move away from its set point. There will be a lot of disturbance, isn't it? So therefore it's not taken as a good feedback. Positive feedback is considered to be a vicious cycle in many cases. For example, second stage of circulatory shock. Second stage of circulatory shock. Look, first stage of circulatory shock is negative feedback. It's dominated by negative feedback. Means all the negative feedback mechanisms try to bring the blood pressure and all the parameters back to normal. But if they fail and the patient progresses to second stage of shock, positive feedback mechanisms begin to operate, and then the patient will not recover by himself or herself. The external treatment will have to be administered. For example, if there is decreased cardiac output, then there is myocardium is depressed because less blood flows in the coronaries, decreased coronary blood flow, and because of that there is decreased venous return and further decrease in the cardiac output. So a vicious cycle develops like this, and change gets enlarged. As I mentioned, in the negative feedback it was error gets minimized. Here the error gets amplified or magnified. But there are certain good examples. I mean, positive feedback is not all that bad. There are some helpful examples of positive feedback in the body. Let's see those helpful examples of positive feedback. For instance, oxytocin in parturition. It's a positive feedback because more and more of the same goes on happening, and eventually the baby is delivered. So you may have read this example that at term the baby starts pressing on the cervix, signals are sent to the posterior pituitary, there is a release of oxytocin, oxytocin comes to the uterus, contraction of the uterus, baby's head is further pressed down, again signals are sent, more oxytocin released, and it goes on and on and on till the baby is delivered. Second example is platelet plug formation or clot formation. Platelet plug formation is positive feedback. I mean, if the vessel is ruptured, initially few platelets come into that area, they adhere to the injured vessel wall and they release certain chemicals. So more platelets will come into that area. They will adhere to the previous platelets. They will release the chemicals and more platelets will come. Finally, they will form a platelet plug and it will stop the bleeding. So again, a good example of positive feedback. Then action potential, depolarization from RMP to threshold. This is by positive feedback. I mean, during depolarization, how it reaches a threshold value is like this. There is initially few sodium channels open. So some sodium enters. Some sodium enters means some positive charges enter, and because of that the membrane potential drops. Membrane potential drops to a less negative value, like from -70 to -68, and that itself opens some more sodium channels. So some more sodium will come in, membrane potential will further drop, and this will go on happening. So it will reach the threshold. That's the positive feedback. Then LH surge leading to ovulation. Another example of positive feedback. Look, during the first few days of the cycle, the estrogen, it exerts a negative feedback on LH. Estrogen keeps the LH level suppressed. But during the mid portion of the cycle, instead of negative feedback, estrogen will start exerting a positive feedback. So, estrogen, LH further secretion of estrogen, and estrogen further LH. So a positive feedback will ensue. This will finally result in LH surge, that's followed by ovulation. So that leads to ovulation. Another example of positive feedback. Then bladder filling and micturition. Act of micturition is said to be a self-regenerative process. It starts as a weak signal, weak process, but it becomes stronger and stronger and stronger. So positive feedback. Head's paradoxical reflex. Head's paradoxical reflex. Well, this is in the respiratory system. Normally we find that there is Herring-Brewer reflexes in the lungs and thoracic cage. That is, over distension of the lungs leads to deflation. So it's a negative feedback which is normally seen in adults. But Head's paradoxical reflex means distension of the lungs leads to more distension, and that leads to further distension of the lungs. So more and more of the same will go on happening. Now that happens at birth. At birth, the lungs are collapsed. They are going to distend after that with each breath, each successive breath, and therefore distension will lead to more distension. That will lead to more distension, more and more further distension. So that's positive feedback, Head's paradoxical reflex. So these are the examples of positive feedback which are helpful to the body. Finally, let's end this section by saying two things. One is that all the positive feedback mechanisms, they end with a negative feedback. It's necessary. I mean, it cannot go on forever. That same thing is happening. It has to end somewhere. I mean, imagine platelet plug formation. If it goes on happening and happening, entire circulatory tree will be filled with platelet plugs and even clots for that matter. So it has to end somewhere. So remember, first point, all positive feedbacks, they end with a negative feedback. Micturition, most of the process is positive feedback. but last signal will be a negative feedback. It will stop the process of maturation, right? So that's one point. And the second point that I need you to understand is that all the positive feedback processes, they can be considered as a part of larger negative feedback process. All the positive feedback processes can be considered as a part of larger negative feedback process. Let me explain how. Look, oxytocin in parturition, it's a positive feedback mechanism that is working for the sake of delivery of the child. Correct. But think about it like this. Here is a female. She has a certain physiology. That physiology was altered due to pregnancy, and with the delivery, that physiology will return to its normal. So, it's a kind of negative feedback. Some change occurred due to pregnancy, and after the delivery, it came back to normal. So, it's a negative feedback. But so parturition itself occurs by positive feedback, but parturition is working for a larger negative feedback process. Or let's say platelet plug formation, it's for stoppage of bleeding. I mean, imagine that there is a circulatory blood volume in the circulation, and because of the rupture of the vessel, the volume is going out, the blood volume is decreasing. Then with platelet plug formation, the bleeding will stop and the blood volume can be restored. So platelet plug formation itself is a positive feedback process, but it is helping a larger process of negative feedback of restoration of blood volume, circulating blood volume. So that is how you should look at these concepts and principles. I mean, that's all for this particular segment of concepts and principles in the general physiology. Next, we will discuss the cell membrane, its composition and transports.